Good afternoon, everyone, and welcome to One World, One Health, One Water. We're happy to have all of you here today, and I see that a few are still entering our Zoom box here. As you enter, could you please follow a few items of Zoom etiquette? Please make sure that you mute your microphone. And please keep it muted until it's time for you to speak, until there's a question and answer period or you're presenting, of course. I want you to know that during the lightning talks, we'll be going through those continuously. You're going to hear a lot about One Health, environmental and human health research going on at Wayne State University. And we're going to go through those continuously. During the presentations, though, please put your questions into the chat box. We expect that you're going to have lots of those, but we have this steamroller going with all these presentations, and we will attempt to answer some of those during the symposium, but we'll also answer some of them after, and those that don't get answered, we will get back to you on. So please do make use of the chat box. Also, to make sure that you have the best viewing experience during this Zoom, please ensure that your thumbnail view is active so that you can see both the presentation screen as well as the speaker. With that, I'd like to kick off One World, One Health, One Water by introducing the Vice President for Research at Wayne State University, Steve Lanier, who has been an incredibly strong supporter of our integrative One Health research. And I believe he's going to give a little piece of good news about the kickoff of this initiative. Steve?
Thank you, Carol. And welcome, everybody. I really appreciate you all giving me a few minutes in this sort of lightning talk session and format. And I appreciate everybody, Delashawn and the team, and Jamie for teeing it up and setting it up, and all the participants for being here. I just wanted to say a few words to sort of set the table. You know, this is my 6th year here in the city of Detroit. And it's probably been one of the most enriching experience that I've had the opportunity to be involved with in my life, the folks and everything that's going on in this great city. One of the things that we've really focused on at the university is to tackle in a real way thematic areas that are relevant to our role as an urban serving university. And the concept has been really to harness strengths, expertise, creative thinking across all disciplines, and assemble them in a way that can have synergism around a topic or a theme that is of particular significance to the communities that we serve here in the city of Detroit. And that's been a journey. And there's various themes that have evolved during this period. But I can't think of any that compare to the groups that have assembled around environmental sciences, healthy urban waters, One Health. It involves folks from School of Law. It involves issues of policy. It involves socioeconomic considerations. It involves public health. It involves our natural environment. It involves the City of Detroit partnerships. It involves Belle Isle. It involves Detroit Zoo. It involves student training, academic majors, and a whole host of talented folks and partners that have assembled around this topic. As Carol and Melissa and Donna and many others caucused around this over the last few years and begin to coalesce these different initiatives, they conceptualized the idea of bringing all these together in a way under One Health. And so one of the purposes of the symposium today is to really launch this One Health initiative. And the journey to get to this point has involved many partners, many leaders, many students, creative thinkers, partners like the Herb Family Foundation, which has been a steady support of our programs in healthy urban waters during this period, greatly appreciated. Partnerships with the city of Detroit, our colleagues in this area, Belle Isle, and others. And many more, I think, that will come out of this session going forward. Recognizing the importance of this topic and the power, I think, of bringing all these talents together, we've launched formally at the university what I call the One Health Initiative. And I've asked Carol Miller and Melissa Runge-Morris to co-direct this. It's a partnership with the College of Engineering, many departments, School of Medicine, Institute of Environmental Health Sciences, Department of Biological Sciences, Department of Chemistry, the Center for Urban Responses to Environmental Stressors. And as you'll hear more from Carol and the team today, we're really excited about this for a number of different reasons. And we've committed some resources to this program to allow it to get out of the starting gate in a big way. And I think that what you'll hear from Carol and the team later is about some money that we've allocated for projects to get cooking, et cetera. I know that Carol and the team, Melissa, have a number of major programmatic grants on the horizon that we're also hopefully will come to fruition and add further momentum to this initiative. So I wanted to, take a few minutes and put this in context and to formally announce the One Health initiative and to convey my appreciation to Carol and Melissa for graciously accepting the task of leading forward, and to thank each of you for engaging and bringing the energy that's really required to realize this vision. So we've carved out some resources, and I look forward to carving out more as we go forward. So unfortunately, I can't hang around with you guys for the lightning talks. But again, thank you so much for the opportunity to be engaged, and I look forward to many, many special outcomes from this session. Thank you, Carol.
Thank you very much, Steve. And I'd just like you to know that we are recording this. So although that you may need to miss the lightning talks, you will be able to listen to them at your leisure if you become available to do so. Again, thank you so much for the support. And just to let everybody know, as you saw on the agenda, we will be having an optional session after the series of lightning talks where we'll, my co-director, Melissa Runge-Morris, and I will be giving a few more details about this One Health initiative that is being kicked off. So that will come after the lightning talks. At this point, I have the pleasure to introduce one of the primary, the president of one of the primary foundations that has been supportive of healthy urban waters. And that's Dr. Neil Hawkins, who is the president of the Fred A. and Barbara M. Herb Family Foundation. As Steve was mentioning, this foundation has been essential to the development and the growth of healthy urban waters and our commitment to the Detroit regional community and in particular the Huron to Erie corridor. The mission of the Fred A. and Barbara M. Herb Family Foundation is to advance an environmentally healthy and culturally vibrant metropolitan Detroit and a flourishing Great Lakes ecosystem. Neil is also an adjunct professor at Wayne State University, where he co-teaches the Sustainable Urban Business Graduate Seminar at the Mike Ilitch School of Business. He holds a bachelor's degree from Georgia Tech and master's and doctoral degrees in public health from Harvard University. So again, Neil, please take our thanks. Thank you for being here, but thank you for the continued support.
Well, Carol, thank you for the welcome. Vice President Lanier, thank you very much for launching this meeting and being with us today. And welcome to all the participants. First off, I want to acknowledge our distinguished chairman, John Erb, who is attending today, as well as Melissa Damashki, who is our Great Lakes Program Officer. And they've been steadfast supporters of this program all along. And it's a terrific program. I would like to just acknowledge that the program so far, the Healthy Urban Waters program, has had tremendous impact, and you've done great work. And at the Herb Family Foundation, we greatly treasure and value that collaboration that we have. But Healthy Urban Waters has been terrific. Today's session looks amazing, so I think you're going to have some great things. But it is extremely exciting, to me especially, to hear about the new One Health initiative. I think bringing together even additional expertise, the great expertise of Wayne State around health is going to be a great service to the region and to the city. So thank you, Vice President Lanier, for taking that step. Now, to all the participants, I wanted to just address you for a minute. I know these are odd circumstances, and I'm sure people get sick of hearing that. The last time I was with you, it was an in-person event. Now we've all become somewhat more accustomed to Zoom events, but it's a reality that we had to do it this way. But that causes me to think about, why are we still able to do this today? Why is there still research to report? Why all of you chose to gather? You know, what is the motivation? And it's really built around your commitment. All of you on this Zoom call, all of the researchers, everyone, you're committed first and foremost, not simply as a job, but this is a mission and a calling for you to take your skills, take your expertise, and help drive better lives for people in our communities, your communities, and deliver more diverse, biodiverse, and healthier ecosystems. So I just want to call out this mission that drives us. You know, if we weren't driven by mission, we may not be on the call today. We may not have delivered the research we wanted to have. And this commitment and motivation you all have, we all have, is about making positive impact. And really, what more important impact can there be than public health? And that's really what you're talking about, individual health and public health, and the links to water, water quality, environment, and other factors in the community. So I really want to thank everybody for your commitment to the health and well-being of all those around us. I want to thank you for your unique contributions through sharing your expertise. All of you, you come from different parts of the research ecosystem, but you choose to apply it to this area. And I really appreciate everybody sticking it out during really what is the worst pandemic in 100 years. And let's hope we keep it to a 100-year pandemic. So in closing, enjoy the meeting. I think it's a unique opportunity. Learn about other challenges that people are dealing with. And really, keep an eye on what's going on that screen and identify colleagues that you can have new collaborations with. I think that Zoom still can be a great place to find new colleagues, learn new things, and really take it to the next level in this new One Health initiative. So congratulations, and thank you, Carol, for having me today.
Thank you, Neil, so much for those comments. I must say the Herb Family Foundation was there for Healthy Urban Waters at the very beginning, about 6 or 7 years ago. And in the interim period, we have received significant funding, additional funding from National Science Foundation, the NIH. Great Lakes Water Authority has been a very strong and steadfast supporter, as well as the Great Lakes Protection Fund. And we have really been able to complete our mission through our collaboration with multiple watershed organizations throughout Metro Detroit. So I wanted again to say there have been a variety of foundations and agencies that have helped support this mission, but Herb was there at the beginning, and the Herb Family Foundation is still there. At this point, I would like to identify and welcome our keynote speaker, Dr. Irene Zagaraki. And Irene is here from Michigan State. Some of you might wonder, well, why are we inviting somebody from Michigan State to be our keynote speaker? This is a Wayne State University event. Well, Irene is a great collaborator for many other researchers here at Wayne State University. And one of the things that we want to be doing as part of One Health, just like we've been doing within Healthy Urban Waters, is to reach out to others and develop new and stronger collaborations. And I think when you listen to the topic of Irene's presentation, and then you listen to some of our lightning talks, you'll see that connection. So Irene, welcome here. Irene is an associate professor of environmental engineering at Michigan State University. She earned her PhD and MS degrees in civil and environmental engineering from the University of Wisconsin-Madison, and her BS degree in environmental sciences while in Greece. Before joining Michigan State University, she held a postdoctoral position at the University of Wisconsin-Madison. Her research is focused on water quality engineering, emphasizing protection of public health and prevention of waterborne disease. In particular, she is interested in microbial contaminants throughout, and their fate and transport throughout the infrastructure of water systems. Her research projects are funded by the National Science Foundation, the EPA, the Department of Homeland Security, and many others. Irene, welcome to this symposium, and thank you for speaking.
Thank you so much, Carol. It's a great pleasure and it's an honor to be here. I'm very excited. This initiative is fantastic, and I do hope that this talk is just only the beginning of our collaboration with my research group and with Michigan State University. It's just the word, one health, one world, one health, one water. It signifies the need for a holistic approach and collaboration. So today, before I go present a case study for a project in Detroit, I want to spend a couple of minutes talking about One Health, like maybe a minute or so. So in the next slide, I'm sure you all are aware of the concept of One Health, which basically the basic idea behind it is that environmental health, human health, and animal health is interconnected. So if you want to design intervention systems, then the higher chance of being successful is if we take into account all the components. And when we identify critical pathways, design surveillance systems, and design intervention systems, we got to take into account the human health, animal health, and environmental health aspect. Now, an example of this might be vector-borne diseases, and how in some cases Scientists were able to control human infections by controlling the population of mosquitoes, right? But for my talk, I want to focus on the environmental and water significance of the One Health approach. So in the next slide here, if we think about a water-focused One Health approach for early detection and prediction of outbreaks, then we see that in terms of viruses, we have viruses that are their exposure is from human to human. We have animal viruses, but we also have direct exposure between animals and humans in terms of zoonotic viruses. Another means of exposure that involves environmental media is waterborne, foodborne, recreational exposure, exposure via vectors that breathe in water, via aerosols, soil, and fomites. So that's an important pathway that we need to take into account. Now, I want to focus on early detection systems and what's the role of water in this case. So if we think about human and animal excrement that eventually ends up in the environment, then if we want to sample the population in terms of define and find community composite sampling locations, a very appropriate medium is water, environmental media. So if we decide to survey environmental media, we may be able to capture the status of population health and also predict upcoming outbreaks. So those locations may be a convenient location in an urban setting is wastewater utilities, municipal wastewater utilities, or it may be hospital wastewater, residential building wastewater, airport wastewater. Now, when we move to rural areas, then it's more complicated to identify those locations since they're not going to be point sources. But we're going to focus on the urban area in this talk. So if we move quickly onto the next slide, the concept of the One Health approach and the role of water and surveillance of water for the prediction of outbreaks, we have described those in 2 publications from my research group that you can, if you're interested, you can just, I have the titles here. And then in particularly, in the next slide, we have a couple of publications where we describe the concept of wastewater-based epidemiology. Now, moving to the next slide, if we want to apply that concept of wastewater-based epidemiology in a urban location, what we claim here is that, of course, the number 1 step is to collect samples and test them, but it's much bigger than this. So there's many components to an approach like this for prediction of outbreaks and surveillance of population health. One is the sampling, of course, but then we also need to take into account many other parameters like environmental factors like precipitation, especially if you have combined sewer overflows, then the fate of those viruses inside the collection system, inside the network, the miles and miles of network, you need to take that into account in terms of lag times and reaction times. And then, of course, strong collaboration with health departments in terms of selecting the appropriate data for correlating with our environmental measurements. So I want to move to the next slide and start talking about an example here of what we've been doing in Detroit. We've been, since 2017, we've been working with the city of Detroit and the wastewater treatment utility, the wastewater reclamation facility, which is the largest single facility treating wastewater in the nation. And it collects wastewater via three main interceptors, the sewer, via sewer lines that then discharged into three main interceptors that end up in the utility. And they capture most of Wayne, Macomb, and Oakland counties. So it's a vast area of networks and that end up in those three interceptors. So in the next slide, then we show those three interceptors and the areas that they correspond to, geographical areas in Wayne, Macomb, and Oakland counties, and then they end up in the water reclamation facility. So in the next slide, then I'd like to spend a minute talking about the partnership. It started in 2017 with funding from the National Science Foundation, and then it evolved, and now it includes a major partner is the Great Lakes Water Authority, which funded us, and we're working closely with them. Also, the partnership involves CDM Smith and Michigan, and Eagle, Michigan Department of Environment, Great Lakes, and Energy. We're still working with the City of Detroit, the health departments, Oakland, Macomb, and Wayne. This is continuing and evolving since 2017. So moving on to the next slide, I mentioned the funding for this ongoing project, which has been from National Science Foundation, Great Lakes Water Authority, CDM Smith, and Eagle. Now, moving on to the next slide, just the next couple of slides shows the sampling. And then I want to start with some of the our results from the initial study that have been published. So moving to the next slide, what we did is we're collecting wastewater samples from the three interceptors that I mentioned above a second ago. And we are collecting, there's different methods where you can collect samples. One can be like grab samples, but what we do is we use, in order to increase the volume of water we sample, we use an electropositive cartridge filter, and we pass the water through it. We discharge the water and we keep that filter, which has viruses attached onto it. Then we bring it into the lab and we elute those viruses out. We do our RNA extraction, DNA extraction, and then we do reverse transcription and quantitative PCR, and we also do shotgun sequencing and Illumina sequencing and bioinformatics for viral diversity. So the next slide shows what we do with sequencing. We start with random amplification, and then we do all these steps. I'm not going to go into detail, but our goal here is to look for unknown, like with the qPCR, we target a particular virus, but with this method here, we target we want to find any virus that may be in our sample, just to find any emerging diseases. So next slide. In the next slide, this, no, excuse me, the previous slide, this one, yes. So using those methods, the sequencing and bioinformatics methods, between 2017 and 19, we surveyed the wastewater from those three interceptors, and we found all sorts of different diseases in the population, viral, we focused on viral diseases, and we found respiratory, enteric, blood-borne, vector-borne, and other viruses. At the time, there was no SARS, but COVID-2, but we found other species of SARS, the common cold viruses, coronaviruses. We found enteric, we found viruses, hepatitis, a lot of hepatitis viruses, and even viruses related to, that may be related to certain cancers, cardioviruses, HIV viruses. We looked at the public health records from the state, and every single viral disease that was reported, we were able to capture through our methods, and we also found more that were some diseases in the excreted by the viruses excreted by the population that the state was not, they were not required to test for. But so this tool proved to be a very important and valuable tool for surveying population health. So in this next slide, I just show that just for confirmation, we did quantitative PCR then for each and every virus just to confirm. So next slide. At the time, there was an hepatitis outbreak, and we were able to capture all 4 species, A, B, C, D, and E. Then next slide. Then we, just to test the predictability of the model, then we looked at hepatitis A. And then in the next slide, we show, this is the epidemic curve. These are the actual clinical cases provided by the state. Then we have sampling here 1, sampling here 2. So we sampled during the peak of the outbreak and then after the outbreak. So the next slide shows here that with the red bars show the measured concentrations of the hepatitis virus that we quantified. And then the blue dots show the confirmed cases about 2 weeks later. So we had very strong correlations 2 weeks ahead of the cases. So that was a good proof of the predictability of this work, of this model. So moving to the next slide, we also focused on herpes virus work, and that's published. And then moving into the next slide, we tested all eight species of herpes viruses. Some of those are very common, so we expected to find them. But there was one species, herpes virus 8, which was really high concentrations. And we were surprised about that, but it ends up being this species is related to Kaposi's sarcoma. It's a form of cancer, which is observed in HIV patients. And we were able to relate that with HIV contigs. So in the next slide, we show we use different methods, and we're able to make that association between herpes virus 8 contigs and DNA sequences and the HIV population in Detroit. So next slide, our next slide now. After COVID-19 became an issue, then we started working with the Great Lakes Water Authority, and we focused our efforts on COVID-19 for an obvious reason, because now that's the biggest problem. So Great Lakes Water Authority has been an incredible partner for this project. So I show here our sampling device, some pictures of our sampling device. And in the next slide, please, These are some pictures of the students, PhD students in the lab, eluting those filters. And then the next slide, now performing RNA extraction and then performing quantitative PCR. And the next slide, please. Those students like looking at this is the amplification curves for the quantitative PCR. Now moving to the next slide, there's another publication that I just show the title here if you're interested, where we describe our methods for SARS-CoV-2 in Detroit wastewater. So next slide, please. Since that publication, we observed, we keep monitoring. So I want to show you some of the latest data. These are the 3 interceptors. Now on the x-axis we show the dates, and on the y-axis we show the concentration of SARS-CoV-2 in genomic copies per liter, and we see how it fluctuates. And we see that in about October we had a second peak that was observed in November. Now, moving to the next slide. This is the next slide, then would be our NIA interceptor. Again, same patterns. We see temporal fluctuation of the concentrations. And then the next slide shows again the third interceptor. Now, we wanted to see if that correlates with our data and if we could have a predictive ability. So the next slide shows some of the data of the clinical cases. Next slide, please. These are the confirmed cases reported by the state, confirmed COVID-19 cases per day for Detroit City, Wayne, Oakland, and Macomb counties. And we see that this is during our period of sampling. We see we had a peak in July and another large peak in November. So now moving to the next slide, please. The blue lines now, the blue bars, correspond to the sum of those clinical cases. So this is the blue bars correspond to the confirmed cases of COVID-19 in the city of Detroit and in Wayne, Macomb, and Oakland counties. On the and they are plotted. They're shown on the axis to the left, confirmed COVID-19 cases. Now on the axis to the right, we plotted the genomic copies per liter of SARS-CoV-2 for the 3 interceptors. Those are the dots, the yellow, orange, and gray dots. And you see that we could see a peak preceding the actual peaks of confirmed cases. Now, these are just the raw data. However, now if we want to really think about how to interpret those results, we need to think about what happened into the system, into the wastewater collection system during sampling. So in Detroit, we have a combined sewer overflow. So when there is a high precipitation event, then we have the storm water mixing with the wastewater, so it dilutes the wastewater, right? So we need to take that into account. So the next slide, in the next slide, I show an example of that, where, for example, for this one interceptor, we have that data for all interceptor, but I'm showing one. The blue line shows the flow of that interceptor, and you see that sometime in June, for example, there's a peak, right? So there was a rainfall event there. So it goes up and down, and then at the same time, the total suspended solids and BOD and nitrogen, they all go up and down because of the dilution issue. So what we did then is we normalized our qPCR data by multiplying by the flow, right?
Irene,
Yeah.
I'm so sorry. I'm so sorry that I have to butt in. You have so much good material, but we're going a good amount over.
Okay, 1 minute. I'll
Thank
wrap
you.
up. Thank you so much thank you for intervening. Okay, next slide. Then we normalized our data. By, and then we also adjusted for population. And now we could see our prediction, which is about a month ahead of, so we were able to predict the peaks a month ahead of time. So going to the next slide. Now we are also surveying the several neighborhoods with different demographics, and we see positive results with every single sample. So we're continuing our centralized monitoring, and we're also doing the interceptors. And that's the end of my talk. Thank you so much.
Irene, thank you so much. You had more material than I think we could test, but we learned a lot. And one thing you're going to see is there's at least one more presentation that actually ties in really, really well with yours.
Okay, great.
And we have some questions for you. I am going to forward those to you, or if you are still around when we're done with the symposium and we have an open question session, I will bring them out then, because I do need to get started with our lightning talks
Thank you,
speakers.
Carol.
Irene, thanks so much.
Thank you.
All right, Barb, I think you're going to be starting the lightning talks.
Very good. Yes, we're going to start the lightning talks with Dr. Tracy Baker, who's going to talk about zebrafish as a model for studying One Health.
All right. Yeah, I was going to try to take over control of the slides, Jamie, but I think I'll just, for time's sake, we'll just do it the way we practice. So good afternoon, everyone. I'm always excited to talk about zebrafish and One Health. Next slide. So zebrafish are an excellent vertebrate species for evaluating health effects of exposure to environmental chemicals and other stressors due to their quick development. As you can see in the time lapse, 24-hour time lapse, which I think we have to advance just once to get that to start. Yeah, okay, thanks. And in 2 to 3 days, they are hatched, all their organ systems are formed and functioning, and they are swimming around. So as you can see, they're transparent, so we can use them to easily visualize development and structures. The life cycle of zebrafish are very short, so in about 3 months, it's from when they're born to when they can reproduce. One female can lay 100 to 200 eggs per week. So that allows large numbers of zebrafish to be obtained for studies. Further, zebrafish embryos, because they are fertilized and develop outside of the body, it makes it easy for manipulation, chemical exposure, as well as observation. Due to their small size, they can be used for high throughput sequencing. So you can see that they can fit well, easily into a 96 well plate. And many adults can fit into one tank. And the system you see here is ours, and we can fit, if we wanted to, about 10,000 fish into just this room. Okay, next slide. So the zebrafish genome is sequenced. And so that and our ability to accelerate genetic studies using knockdown and overexpression have really led to the wide use of zebrafish. So due to genetic, developmental, and organ system similarities with humans, the National Institute of Health, as well as many others, accept them as a model for human development and disease. And so here's a very short list of some of the areas of study that have been utilized. Next slide. So here you can see the genetic overlap with humans, mouse, and chickens, and next, other animals. So this allows us to translate results in zebrafish not only to humans, but also to pets and wildlife. Next. And the study of conservation of genes also shows that they're closely related to other fish species, such as rainbow trout and gar. So that allows us to evaluate potential health effects in wild fish in the Great Lakes. And next, so some of our other cool applications in the zebrafish lab, which you will see some of these later. We can inject and grow fluorescent cells and other cancer cells and watch that in a zebrafish. We can make fluorescent transgenics, observe other fluorescent particles like nanoplastics and microplastics, look at developmental abnormalities. We can also do behavior analysis. So using this device you see in the middle of the screen, the camera and the software track the fish in 24 well plates, and we watch as they respond to light and dark. And so usually they'll have no movement in the light and quite a bit of movement in the dark. And so this allows us to evaluate hyper and hypoactivity as well as study neurodevelopment. We can look at the microbiome and changes, gene expression, pathway analysis, and do all of that even at a single cell resolution.
Thank you, Dr. Baker. Next, we'll hear from Sadaf Tiamori talking about impact of storm sewer networks on urban groundwater.
Thank you, Barb. Hello, everyone. This is Sadaf, and I'm gonna talk about how storm sewer networks can affect the urban groundwater using the results from a groundwater flow model. Next slide, please. This slide shows the position of Detroit City in southeast Michigan relative to 4 watersheds of the Rouge River, Ecorse Creek, Lake St. Clair Drainage, and Clinton River. These 4 watersheds are identified as the geological region and forms the basis and boundaries of the groundwater flow model. Next slide. For creating the regional groundwater flow model, we collected many data sets, including precipitation, evapotranspiration, hydraulic conductivity of the soil, the boundary conditions, pumping wells, lakes, rivers, and storm sewer net lines, which is basically a combined sewer system in southeast Michigan, and then applied all of this data into the groundwater grid in the mud flow environment, then run the model and get the results. The results show groundwater flows from the northwest to the southeast and discharges into the Detroit River. Also, we have a shallow groundwater which can be found at the depth ranging from 3 to 10 feet below the surface. Next slide, please. This slide shows the overflow risk of a combined sewer system. The combined sewer system in the urban areas can reduce the groundwater recharge. The conductance of these sewer lines plays an important role in the combined sewer overflow events and the distribution of storm water in the region. In this figure, you can see the risk of the combined sewer overflows in the Metro Detroit area. From this figure, we can conclude that as the conductance of storm sewer lines decreases, probability due to the aging of the network and further deposition of the sediments in the pipes, the overflow risk of sewer system will increase in the northwest of the Metro Detroit area. Next slide. And this is the groundwater flow model for a small area in the southeast side of the Detroit City called Recovery Park. In its local scale model, the results show that this sewer line system changed the groundwater direction, that is a groundwater flow in the opposite of the original flow direction. So we can say this sewer line system as one of the urban settings have a large influence on localized as well as the regional groundwater flow patterns and combined sewer overflow events. Thank you.
Thank you, Sadaf. Next, we will hear from Dr. Rodrigo Fernandez Valdivia talking about decoding PFAS exposure in breast and lung carcinogenesis.
Thank you. Thank you. Next slide, please. We'll go really fast on this. So the results we're presenting is about PFAS exposure on either primary cells as well as in mice. And our interest is to understand how these chemicals impact normal function, normal biology, as well as how they lead to carcinogenesis in both breast and lung. So here what we have is some results from exposure of lung primary cells to two PFAS compounds, PFOS and PFOA. With both of them in A, you can see induced transcriptional reprogramming. In green light, you can see the genes that are upregulated, and in light blue are the genes that are downregulated. Two examples of those are shown in B, where you can see the genes like CYP26B, a gene that's upregulated by PFOS treatment, and GRN is one of the genes that is downregulated in the case of PFOA treatment. Remarkably, treatment with either PFOS or PFAS, as you can see in C, induce a strong response to chemicals, response to stress, as well as pathways involved in developmental processes. And we know developmental processes are also critical for carcinogenesis. Next slide, please. So in the previous slide, we were showing the transcriptional reprogramming in normal lung primary cells. And what we see in the case of lung cancer cells, or malignant cells here, is that in A and B, we can see that both PFOS and PFOA induce cell proliferation. And in C and in D, you can see that that increased cell proliferation is caused due to reduction in the G1 phase of the cell cycle, which it will be the area under the curve of the first one. So next slide. You can see also in the case of mammary gland cells, both PFOS and PFOA also induce transcriptional reprogramming. We have genes that are upregulated and downregulated. And similarly, we see that genes that are, or pathways that are involved in response to stress, as well as pathways involved in development, are involved in this process. Next slide, please. So, but this is one of the latest results that we have. Strikingly, what we have seen is PFOA particularly is extremely toxic. This data comes from animals that were exposed to PFOA for 5 days only at 25 milligrams per kilogram per day. And you can see both in males and females, there's a dramatic decrease of weight loss. So we have seen also that this particularly affects the mammary gland fat pad. We have a significant reduction. The data is data that we are processing right now. It's a very important finding in the case of PFOA in particular. It's extremely toxic PFAS compound. Thank you.
Thank you, Dr. Rodrigo. We will hear next from Kathleen Sexton on community partnerships for watershed education.
All right, thank you. So today I'm going to be talking about community partnerships for watershed education, and that ties in really well with the One World, One Health, One Water theme. Next slide. So a lot of you guys probably already know who we are, but just in case, I'm going to go into a quick background. We are an environmental nonprofit, and we're based in Rochester Hills, but we cover the entire service area of the Clinton River watershed. So it's 760 square miles, 75 different communities are included in the watershed proper, as well as the direct drainage communities. And our mission is to protect, enhance, and celebrate the Clinton River, its watershed, and Lake St. Clair. We do that through our main program areas of education, stewardship, and watershed management. Next slide, please. So the Clinton River watershed is a it's a densely populated urban watershed. And so we rely on a lot of different partners to do what we do. And I'm going to focus on our education component of that today. So firstly, our public education plan, that's a strategic document that outlines our commitment to public education in the watershed. So engaging the community and watershed residents in watershed education, stormwater, and water quality. We do that through our citizen science programs, which are things like Adopt a Stream and Stonefly, where we get people out in the river, out in the watershed, out in different lakes and surface waters that you guys are probably familiar with. And we get them collecting benthic macroinvertebrates, collecting water quality data, things like that, as well as our Stream Leaders program, where we get students out in the watershed collecting the same type of data. Analyzing that data, reporting it to us. We have about 20 years of data based from our Stream Leaders program, as well as grant projects that we focus on restoration and green infrastructure and things like that, or like the one that I'm going to go into a little more detail, the Wayne State University microplastics grant project that we partner with them on. Next slide, please. So focusing on the One Water initiative, we are a small nonprofit and our budget is just as small. So we really work, we rely on our community partnerships in order to be able to do what we do. So these are just some of the partners that we work with: other watershed groups, other nonprofits, universities, community foundations, and our communities and school districts. Next slide. So some of the outcomes of our recent projects: we've had almost 800 students from Lakeview Community Schools visit and tour the Heart Lab at Lake St. Clair Metro Park. We've been able to network with various stakeholders through the microplastics grant with Wayne State University. We've been able to host those publications on our website, send them out to multiple communities within the watershed that we contract with, create and distribute single-use plastic and green stormwater infrastructure flyers that are that correlate with the project goals, and also simple things like stewardship and community engagement within our watershed. So we wouldn't be able to do what we do without all the partnerships. Thank you.
Thank you, Kathleen. Our next lightning talk comes from Dr. Bill Schuster, who's going to talk about dorm level wastewater monitoring for SARS-CoV-2. Thank you.
Thanks so much, Barb. Let's see. So I don't see the presentation.
Jamie?
Here we go.
There we go. Good deal. Thanks, folks. And this study is reflective of the One Health, One Water perspective. And let's go to the next slide. We had a pulls together an interdisciplinary group of people to take this from all the angles of what was essentially wastewater-based epidemiology. Next slide. So once we figured out as a community of practice, us researchers, the SARS-CoV-2 material sheds in fecal matter and flows in our extensive and contiguous wastewater collection and conveyance system, we then had a forum to monitor for this virus, and it's a leading indicator to predict the spread, as pointed out by Irene earlier. And so this is where Wayne State engages in wastewater-based epidemiology and the potential to focus testing and resources in a larger public health forum around driving down any kind of spread. And so we were looking primarily at congregate housing. It's a particularly important venue for community spread, and it's protective of our Wayne State dorm residents, but also of the greater downtown community. because we do some monitoring around Harper Hospital and other sewer sheds in the area. And so we joined a statewide network. This was a big part of getting the funding to do this work and to track and study the dynamics of SARS-CoV-2 viral material as a tracer for spread. And so using state-of-the-art equipment through this grant program and a shared consistent protocol, we've joined with our, this group of researchers and health departments around here to really focus in on how does this dynamic operate. So let's go to the next slide. And so some example data here. I'll ask you to focus in the middle on the PM-MOV, that's the pepper mild mottled virus. That confirms that we're actually seeing fecal matter. That's a fecal matter tracer. So that's a check. On November 12th, 2020, that left-hand column, what we're looking at in University Towers, we paired that, we have a strong SARS-CoV-2 signal paired with at least one individual in quarantine in the University Towers dorm. When we moved to 11/19, November 19th, 2020, we still have a confirmation of that fecal matter signal through the PM-MOV, but we've declined over that that succeeding week where we have no individuals in quarantine or isolation in that dorm. And respectively, right there, we know that we have some correspondence between SARS-CoV-2 signal and the number of people who are basically pre-spiking our dorms with SARS-CoV-2 signal. Next slide. And this is just a quick wrap up that you can take in on the recording. But this is a retrospective study that we are endeavoring to continue and also to help. Let's get some good time series models here going to understand sources of uncertainty and moving forward, better predictive ability of these leading indicators. Thank you.
Thank you, Dr. Bill. Up next is Dr. Adrian Vask talking about environmental health assessments using biodiversity of water mites.
Hi, everyone. Thanks for having me. So I'll talk to you briefly on the water mites that I work with, which are aquatic arachnids that are found in freshwater habitats around the world. Can go to the next slide. And these are interesting to us because they're known to parasitize and feed on mosquitoes, on mosquito larvae, and parasitize adult mosquitoes. So there's a huge economic and environmental service that they provide that we still don't know much about. So I collected water mites from the habitats around Detroit and at the Lake Sinclair Metro Park, and I fed them mosquito larvae. So you can go to the next slide. And I looked at several different kinds of water mites, and I checked if these different kinds of water mites would actually feed on mosquito larvae, and I found two water mite types. On the left, you see predators, Limnesia and Hygrobates. I observed feeding on water mite, feeding on mosquito larvae, whilst these other kinds, Pneumania, Hydracna, Oxus, I did not see preying on these mosquito larvae. And this is the first time that this has been done systematically in the lab. So you can go to the next slide. And here we have some documentation of these water mite types feeding on mosquito larvae. On the left, Limnesia, and on the right, Hygrobates, and you can just see the mite there on top of the larvae. You can go to the next slide. And we got this data by observation and by molecular analysis. So we use the same techniques that were mentioned in the other talks on detecting viruses and these other things. And we were detecting mosquito DNA inside of the mites. And we found all these different kinds of mosquitoes. And we showed 2 new mosquito larva predators. Thank you.
Thank you, Dr. Adrian. Our next lightning talk comes from Danielle Meyer. She will talk about the accumulation of potential health effects on nanoplastics, excuse me.
Hi, everyone. Thanks for having me. Today, I'll be discussing the Baker Lab Nanoplastics project. Next slide, please. Nanoplastics are small pieces of plastic waste that have been released into the environment as a byproduct of personal care products, as well as larger plastic waste bits that get broken down through environmental processes. Now, because these plastics are not efficiently removed through wastewater treatment, they're found throughout fresh and saltwater sources and have been detected in at least 90% of bottled and tap water in the US. So we know from this that humans and aquatic animals are frequently exposed to nanoplastics, but there's still a lot of work to do on characterizing the possible negative health effects. Next slide, please. So we used the larval zebrafish model to study the effects of exposure to 2 different sizes of nanoplastics, 50 and 200 nanometers, at a range of environmentally relevant concentrations. We exposed the fish for the first 5 days of life, looking for different signs of toxicity on day 5, including if and where nanoplastics are accumulating in the developing fish, the light-dark activity of the fish, and any genomic changes due to nanoplastic exposure. Next slide, please. First, we looked at accumulation in the body. Some of the nanoplastics we use have green fluorescence, so we can track where they accumulate in the fish. Here at the top left, we can see as the concentration increases, an increase in nanoplastics in the liver and GI tract, and a little bit around the head. Now we've just started to look more closely at the brains, and here at the bottom right, you can see some of the green nanoplastics accumulating near blood vessels stained in the brain. Another sign of toxicity is changes in neuromotor activity. We study how the fish respond to a series of alternating 3-minute light and dark intervals. You can see here that larval zebrafish move less in the light and more in the dark intervals, and any change in this natural pattern indicates a toxic response. Although our smallest nanoplastics don't seem to have an effect, the 200 nanometer plastics are showing a hyperactive response at higher concentrations. So overall, these plastics seem to be accumulating near the brain and are affecting neurobehavior. Next slide, please. We also looked at how these plastics are changing gene expression. We saw very few changes due to the smaller plastics, but the larger size affected neurodevelopmental and neuromotor pathways, as well as hepatic and cardiac system function, pretty consistently across the higher concentrations. Overall, effects on motor function, brain development, and energy metabolism make sense with the changes in activity levels we're seeing, and it will be interesting to look at the health and reproductive fitness of these fish as adults. Thank you very much to Anna Maria Petrive, Adam Peterson, Abraham Soto, and everyone else for their hard work contributed to this project. Thank you.
Thank you, Danielle. Our next lightning talk comes from Dr. Tam Perry, and we'll be talking about the perspective of the changing environment from older Detroiters.
Hi, everyone. Thank you so much. Are my slides coming? Okay, good. Well, I will say our project is sort of slower to start because of COVID, but just to be clear what the project is and aims to do. So it's really to document older Detroiters' experiences of belonging and sort of to understand their different perceptions as our environment changes. So one is to understand older adults' perceived vulnerability to climate change. 2 is to perceive challenges that they're currently facing and environmental justice concerns. And 3, to actually develop a pool of advocates and research participants for future studies on seniors' vulnerability to understanding changing environments. So it's also a way to sort of use older Detroiters as a resource in environmental research. Next slide, please. So what we spent time doing is assembling who is going to be on our team. So we actually have 2 consultants on the grant that are seniors themselves. And then we also have a student assistant, and my project director, Evan, is on this call right now. In addition, we spent the summer developing questions that would really be understood by older residents in the city. So we took a lot of time developing questions and then checking them again. Also, the way we developed our consent materials and how we explained the project in terms. We actually then had numerous local environmental groups, staff members look at and examine our research questions. Interview questions, in order to, again, make sure that we were in line with both the key issues on environment facing Detroit, as well as sort of an ability to ask it in ways that could be understandable to older Detroiters. Next slide, please. So I would just say that thus far, the whole study of doing focus groups, interviews, and a survey is sitting with the Wayne State IRB, and I know that that there are a lot of delays due to COVID with that work. So I would say the other thing we did is we developed a handout that we're going to give everyone who participates in our study that actually shows environmental resources. I'm happy to share that with anyone on the call once it's approved by the IRB. So I guess what to leave you with is that we are trying to do a project that is participatory, to use best practices on ways to engage older adults as we move forward in this project. Thank you.
Thank you, Dr. Perry. The next talk comes from Dr. Paul Kilgore, and he will talk about infectious diseases and One Health, the impact on Michigan.
Thank you very much. And I just wanted to say thank you very much to Carol, Jamie, Barb, Bianca, and the whole team, and really appreciate this invite. We can go to the next slide. So one of the things that we know in Michigan is that we have zoonotic diseases. They're actually in many, many counties across Michigan. One of the things that we're very concerned about, of course, is the growing rate of disease that we're seeing. And some of those I'll talk about today in a very limited time. So go ahead, next slide, please. One of the things I did want to point out too as well is that one of the things that we see from the epidemiology of these vector-borne diseases is that it is occurring statewide in counties throughout, especially the lower peninsula, but also some of the upper. Also, we know that they do have a disproportionate impact in younger children, especially as well in older adults. We see severe clinical disease. Also those that are immunocompromised and pregnant women as well. And one of the interesting things about what we know currently, not only nationwide, but in Michigan, is that as these diseases are evolving, we're beginning to realize that we have not only a dearth of data and disease burden information, but also we don't really have great tools widely available for treatment and prevention, especially in the realm of vaccines. And let's go to the next slide. So one of the things I wanted to highlight today is the growing burden of West Nile. So since 2014, we've actually seen an increase. And in 2018, in Michigan, there were actually a total number of 144. This was preliminary data in Michigan. But the final tally in Michigan was 144 cases of West Nile disease. And I think this is really important to realize, especially as we think about the changes in habitats, the changes in exposure for potential animals that we're going to be exposed to. So deer are very important in some of these diseases. When we think about exposure to West Nile, we have the vectors already in Michigan for this. We have Culex species, but we also have other important mosquito species, particularly some of the Aedes aegypti and other important species that we think about with other vector-borne diseases. I highlight West Nile virus because we're actually seeing that as the #1 vector-borne disease in Michigan, and its range is expanding. Of course, we do see expansion in other diseases that are tick-borne, especially Lyme disease, which has been quite dramatic in its expansion across lower Michigan. But one of the reasons why West Nile is important is that we have that as our #1 problem, and we're really focused on that in Michigan State surveillance. And next slide, please. So I just wanted to end up by pointing out that when we look at the disease burden across Michigan, we realize that we have not only animals where we can detect these important vectors, but also we can actually do testing on animals and in humans. And I'm not sure if people have realized last year during the pandemic in July, we had some cases of Jamestown Canyon virus disease. That virus had actually been identified way back in the 1980s in Michigan, and we knew that it was circulating. We knew that people were being exposed to these viruses, but now we're starting to see more clinical disease. And my prediction is in the next 10 years, we're going to see growth in vector-borne disease. So the need for improved arbovirus surveillance and also measurement of disease burden and understanding who's impacted by these diseases is really going to become even more important going forward, especially as our population ages and especially as some of the habitats are shrinking for some of the animals. And we have deer populations that have always been large in Michigan, but we know have been expanding in some counties across Michigan. Also, we're seeing new reports from other states with the vectors that can actually carry and transmit the viruses. So that activity in cooperation with CDC is going to be extremely important. So thank you very much.
Thank you, Dr. Kilgore. Next, we were going to hear from Dr. Carol Miller.
Yes, thank you, Barb, and thank you so much to all of our speakers. I have truly been enjoying this and find it very energizing and exciting. I am going to cede my time. We are a little bit over. Jamie, you can just perhaps run through the slides so people get an idea that the presentation was dealing with phytoplankton measurements at the Waterworks plant of the Great Lakes Water Authority. And in this project, we are looking at historical changes in phytoplankton diversity and density. And the point that I was trying to make in this presentation, I didn't really have any final conclusions because this is work that we're starting to do to look at a very long trends of this phytoplankton data. But the point here was that there is a lot of existing data out there. We don't always have to put in new sensors. We don't always have to spend the millions of dollars to start evaluating a new parameter. Sometimes it's very useful to look at existing data, long-term data that we have. And we have that for the Detroit River in terms of flows and in terms of lots of information regarding temperature changes, water levels, phytoplankton, and so on and so forth. The last slide, Jamie, if you could put that up, was simply to say that I think this topic that I was starting to discuss about regarding using existing data, historical observations, and so on ties in really well with another sort of way of looking at things, and that's using traditional ecological knowledge, which is something that First Nations has really developed into a science of its own. And I wanted to promote an upcoming seminar dealing with TEK, and this is from the T-Rust group. And I will now move to the next speaker. And again, thanks for everybody that's participating.
Thank you, Dr. Miller. Our next speaker on Lightning Talks will be Dr. Bridget Baker, Mixtures of Emerging Contaminants in the Detroit River.
Thanks, Barb. Just wait for my first slide to pop up. Okay, so yep, I'll be talking about contaminants of emerging concern, which are a diverse group of chemicals that include pharmaceuticals, personal care products, pesticides, and PFAS. Next slide, please. And our study on CECs was performed within the Lake Huron to Erie corridor centered around Detroit. We collected composite surface water samples biannually over 2 years at 6 locations and analyzed these samples for over 150 pesticide, PPCP, and PFAS compounds. A total of 50 compounds were detected at parts per trillion levels. And the number of detected compounds was significantly higher downstream as you get towards the Rouge River mouth and the Detroit River International Wildlife Refuge. Next slide, please. So in addition to the number of compounds, cumulative compound concentration also increased downstream. Synthetic sweeteners accounted for the greatest percent of cumulative concentration, followed by pesticides, pharmaceuticals, and then stimulants. Interestingly, pesticides were more prevalent at the northern end of the study area in Lake St. Clair, while synthetic sweeteners and pharmaceuticals were more prevalent downstream. Next slide, please. So the 14 bolded compounds on this table are those that we consistently detected across sampling events. And I'll go through talking about some of these. So atrazine contaminated source waters for drinking water treatment plants like the Detroit River are of concern due to risks for children from combined exposure via drinking water, food, and residential landscaping. Studies have also shown impaired survival, growth, and reproduction in aquatic species. And atrazine is a known endocrine disrupting compound that does exhibit toxicity at concentrations below established water quality benchmarks, particularly when exposure occurs during development. The other herbicide we consistently detected was 2,4-D, which is also particularly toxic to fish, but due to a short half-life in aquatic environments is considered safe for aquatic species. However, studies that investigate environmentally relevant concentrations are scarce. And like atrazine, 2,4-D is a known endocrine disrupting compound. For the synthetic sweeteners, ACE, K, and sucralose, research in aquatic species has revealed changes in gene expression, oxidative stress, microbiome, and behavior. And also, their transformation products have potentially greater toxicity in aquatic ecosystems, but remains understudied. Caffeine exposure can result in developmental toxicity and anxiety-like behavior in aquatic organisms. And interestingly, this is exacerbated by co-exposure with the synthetic sweeteners. So this underscores the potential that lower levels of contaminants in mixtures can induce more severe outcomes than individual contaminants alone. Environmentally relevant acetaminophen concentrations are associated with oxidative damage as well as reproductive, neurobehavioral, and transgenerational toxicity in freshwater organisms. And the persistence of antibiotics like sulfamethoxazole in aquatic environments, even at low levels, and particularly within urban water systems, drive antibiotic resistance. Ecotoxicology of the remaining human and veterinary pharmaceuticals here is understudied and typically lack assessment of non-standard endpoints, non-target organisms, and environmentally relevant exposure paradigms. And I just want to briefly also highlight that PFAS compounds were also detected as mixtures. Next slide, please. In summary, detection of mixtures for all compounds was the norm. All of these results are informing environmentally relevant follow-up studies, both in the lab and the field. And thanks to Alex Heimbaugh, Gianluca Speroni, Destiny Johnson, Tracy Baker, other water lab members, past and present, and the U.S. Fish and Wildlife Service for help in completing this project.
Thank you, Dr. Baker. We'll hear next from Scott Carter from the Detroit Zoological Society and their One Health role.
Great, thank you. Thanks to Carol for inviting me to talk. We always like the opportunity to talk about what we're doing. We still are kind of surprised that many people don't realize what the science and research programs are for the DZS and for many zoos, actually. Next slide, please. We are engaged on 2 campuses currently, the Belle Isle Nature Center on Belle Isle, and of course the Detroit Zoo in Royal Oak. In a normal year, we are somewhere around 1.4 million people through our gates, counting both campuses every year. So we have the potential to engage with millions of people over, decades of time. Next slide, please. We have a field program, a field research program with work that we are either doing or supporting on 6 continents currently. Work that we are doing ourselves. Local work includes work in the Detroit River, studying mud puppies, a kind of underappreciated and often persecuted native amphibian that we've been tracking for probably close to 10 years now, through primarily census work with mark recapture over many years. We've also recently begun work on an animal health project in the Falkland Islands looking at penguins. Those of you who are familiar with the zoo know how important penguins are to us. We have a study site in the Falkland Islands looking at 2 populations having, right before the pandemic, really just began a project looking at health, primarily disease issues of the free-living penguins, and also contaminants because of a growing oil industry around the Falkland Islands. Many of our other projects are dealing with either captive breeding and release, which is primarily of amphibians, and we have less of a kind of a health or anything beyond kind of a population approach to that work. But I would also mention here, although it is not part of the work I'm talking about today, but our on-site work is very heavily focused on animal welfare. So our endocrinology lab is dedicated completely to animal welfare. Next slide, please. Our veterinarians are especially focused on a One Health approach. Many of the diseases that have been talked about by Dr. Kilgore and others today, like West Nile virus, rabies, these are diseases that we have to be constantly vigilant about. And of course, they're reportable to the state. And we are always engaged with the state when things like this are happening. But also for species that are don't have a known or obvious human link, like bee virus, or sorry, not bee virus, chytrid fungus, both in frogs, which of course is in Michigan, and of course the new bee sow, which apparently is not in Michigan yet, but we're all watching for it. Next slide, please. And then I wanted to close just again, making the point about the potential reach we have with over 1 million visitors a year and the opportunities that we have to engage people on issues, especially around One Health. Again, I thank you for the opportunity.
Thank you, Scott. Our next speaker will be Clayton Hayes, speaking on the Rouge River Archives.
Hi, everybody, and thanks, Barb. I wanted to mention at the start that the few slides that I have are an adaptation of a presentation that Barb and I gave to a local engineering group. So, some credit to her, most credit to her, to be honest, for the content here. But my name is Clayton Hayes. I'm a librarian at Wayne State, and I work with the College of Engineering. Next slide, please. The Rouge River Archives is a collection of documents produced by Rouge River related projects from about the mid-50s to the mid-90s. Reports, guides, strategies, remediation plans. There's some monitoring data in there as well. And this really originated with Dr. Ralph Kumler, who was a key figure in starting the civil and environmental engineering department in the College of Engineering. And there's material from other researchers from CEE, as well as biosciences, chemistry, geology, medicine, pharmacy. It's kind of all over the place. It's a lot of different stuff in there. And it was moved into the library from the environmental engineering lab, and it's been completely cataloged and it's in our collections. So next slide, please. You can get to it by going to the library homepage, library.wayne.edu, or to our catalog page, elibrary.wayne.edu, searching for Rouge River Archiving quotes, and then combining that with other terms like authors or subjects or anything like that. You can contact myself or Barb for more information about this. Next slide, please. The building in which it's housed at the moment, or the building in which it's housed, the Purdy Kresge Library, at the moment is not open to the public because of COVID. It's one of the libraries that we shut down while this is all going on. But I've been told that access to the archives is available by appointment. Haven't had anybody take us up on that just yet, but it is available. So if you are interested, you don't have to be a Wayne State affiliate in any way. It's open to the public. You just need to contact myself or Barb, and we'll try to make arrangements to set that up. And we can give you more information about what's in there as well. And I think that that's it. Thank you.
All right. Very good, Clayton. Thank you. Jessica Phillips will be talking to us next on occurrence of pharmaceuticals and emerging contaminants in Detroit water bodies.
All right, thank you for having me. I'm a student in Dr. Tracy Baker's water lab at Wayne State, and I'll be talking about pharmaceuticals in the Detroit water bodies and health effects of fish living in these waters. So next slide, please. So as Dr. Bridget Baker mentioned previously, there's numerous contaminants that have been found in Detroit water bodies, including numerous pharmaceuticals, such as your anti-diabetic drug metformin, as well as antihypertensive agents that affect the adrenergic system, anti-epileptics, and others listed on this slide. With the amount of prescriptions increasing in the US, 500 million alone in the last decade to 4.2 billion prescribed in 2019, this is a problem that we will see continue to grow. Therefore, we began a study at one of the intake plants providing water for the residents of Detroit, highlighted here, with two aims in mind. First, to determine the short and long-term effects of environmental endocrine disrupting chemical, or EDC, exposures, as well as any transcriptomic changes that may occur. So next slide, please. Previous study in Dr. Baker's lab have shown a wide range of responses to EDC exposure. From phenotypical abnormalities in a previous metformin study, we found increased incidence of deflated swim bladders, as well as yolk sac edema with metformin exposure, hypoactive behavioral changes, and transcriptomic changes in zebrafish. So with this in mind, we set up a lab at the Detroit pilot plant where that water is brought in from the Detroit River to become tap water and raise zebrafish in either raw, untreated river water, which you can see being pumped into the system here, or in a separate reverse osmosis water system. Now we began this experiment with 4 groups of fish born in different months with endpoints of abnormalities, behavioral changes, and transcriptomic changes in their larval offsprings as our endpoints. We also sampled the water monthly in order to track chemical exposures. So next slide, please. So one of the main endpoints we chose to focus on was fertility. And as you can see here, the raw water, otherwise known as System A, produced significantly more eggs per female than the reverse osmosis group, or System B, as seen in the top graph. However, 23% of the eggs in that System A were already unviable at less than 1 day post-fertilization versus only 18% in the controlled fish. The raw river water also produced a larger number of alive but unhatched eggs at our endpoint, which was 5 days post-fertilization, at around 20% versus just 17% for the control. And additionally, eggs that we plated and raised in the incubator for each system, raw river water had a much higher mortality rate of 77.5% compared to only 74% for the reverse osmosis water. So next slide, please. In terms of abnormality, the data was really dependent on the month of the parent's birth and when they were added to the system. However, when the groups were combined overall, total abnormalities were more significant in that raw river System A water. And several abnormalities were approaching significance, such as uninflated swim bladder, yolk sac edema, and the presence of multiple abnormalities in the offspring as seen in these pictures. Now, no behavioral differences were seen, and we are still isolating RNA to evaluate transcriptomic changes in brain, liver, and gonadal tissue. However, these preliminary results are concerning, as the Detroit River is a major water source for local wildlife, as well as over 4 million inhabitants of the Detroit metro area. So thank you to Dr. Tracy Baker and her lab for all your assistance with this research.
Thank you, Jessica. Next, we'll hear from Zoha Siddiqa, evaluating petroleum-derived VOCs.
Thank you, Bar. Thank you, Bob. Hi, I am Zoha, a third-year doctoral student in the Pharmaceutical Sciences program, and I will be introducing my research on the effect of petroleum-derived VOCs on Daphnia pulex. Next slide, please. VOCs are volatile organic compounds, have high vapor pressure and low boiling temperatures, and exert adverse effects on the respiratory, circulatory, and the nervous systems. So our research is focused on a petroleum-derived VOC, BTEX, that is now associated with preterm births and miscarriages. As VOCs volatilize at relatively low temperatures, it is challenging to elucidate their adverse effects. But we hypothesize that risk resulting from exposure to VOC mixtures can be estimated using the concentration addition model. Next slide, please. To prove our hypothesis, we developed a stainless steel exposure chamber that has a glass top and bottom and stainless steel inlet and outlet pipes. The VOC is stored in a glass container with a nitrogen atmosphere, and it is infused into the chamber through a peristaltic pump. A telecentric lens takes a 5-second video every 5 minutes, and these videos are analyzed for the distance traveled and the angle turned by the organism. Next slide, please. This slide shows the results from toluene exposure to Daphnia pulex. Daphnia pulex was exposed to an increasing concentration of toluene from 1 part per million to 25 parts per million for 4 hours, where the organisms were exposed to each concentration for an hour. As seen in the graphs A and B, as the concentration was increased, the distance traveled by the Daphnia was decreased significantly and the angle they turned increased significantly. These are the toxic endpoints for Daphnia. We ran another set of exposures, those are graphs C and D, where the organisms were continuously exposed to the control media and 5 parts per million of toluene for 4 hours, but we did not see any effect. So this illustrates that the significant effect we saw in our previous study was due to the 25 parts per million of toluene, which leads us to deduce that the lowest observed adverse effect level for toluene is in between 5 parts per million and 25 parts per million. Similarly, we plan to determine the lowest adverse effect levels for the remaining BTEX chemicals and apply them to evaluate the toxicity of BTEX mixture using the concentration addition model. Next slide, please. Lastly, I would like to thank all the different departments and the PIs associated with this project. Thank you.
Thank you, Zoa. Dr. John Norton will talk to us next on assessing sustainability trade-offs in the water sector.
All right. Well, thank you very much for this opportunity here. It's been a really a delight to hear all of the other talks, and quite frankly, gosh, geez, some of them are just fascinating. I wish I could have asked a dozen questions. I'm sure that's the way with everybody. I am the Director of Energy Research and Innovation for the Great Lakes Water Authority. We actually treat all this water. We treat water for about just over 40% of the state of Michigan, and we treat about 30% of the state of Michigan's wastewater. When we're looking at improving things in general, and well, let's do this or let's do this, there's always a trade-off. There are always trade-offs. It's amazing to me that I've gotten into some amazingly challenging discussions with folks who actually insist that that's not true. They're wrong, unfortunately. If you got more in some area, you get less in another. You can try to maximize it in all cases, of course, which is what we are always trying to do with our research and innovation, but it's not always the case. Here's an article here you see on the right side of the screen that talks about the COVID-19 surface disinfectants polluting indoor, causing indoor air pollution. Not saying that we shouldn't do one or the other, but just to point out that that is a reality of the situation. Despite the, and I have the queen, the rock root queen, I want it all, which is I want it all and I want it now. That's not the case. That's not the reality of the world. But interestingly, there are some very good, I'll say recently, but within the last 20 or 30 years, some very good methodologies for trying to assess trade-offs, trying to help the discussion improve when you're looking at one thing versus another. Next slide. I wanted to give like an example of some of the trade-offs that we're faced with. So water use efficiency, very important, right? I probably get asked several times a week in my role as a director for the GLWA about water efficiency and water efficiency savings sorts of things. And my result is, oh my gosh, we have the world's largest freshwater source. We're treating that water, we're making it better. We're then treating it again at the wastewater treatment plant, and it's going out to back to the river cleaner than the river itself. At the board chart conference at the University of Michigan earlier last year, the winner of the board chart award for outstanding research was Michelle Prevost, who has done work for the last 25 years looking at water use efficiency and the role of water efficiency use and sustainability measures in hospitals as a cause of lead and copper contamination, as well as the rise of opportunistic pathogens. It's absolutely the science. It is there. You do this, you have a little trickle of water flow coming out, the water will age and thus be worse. Carbon emissions. You want greenhouse gas emissions to reduce? Fine. Nuclear power has the lowest embodied carbon emission per energy methodology. I used to think it was hydroelectric, but it turns out that hydroelectric can actually produce a huge amount of greenhouse gas emissions. Who knew? Turns out that it gathers up, it lets the organics settle out very deeply. They degrade. Instead of forming CO2, they form methane, and this is very bad. There we go. There's a whole list of other things. Another example here in the region is when we dosed orthophosphate to our water to prevent or inhibit lead leaching back into the water. We then at the tail end at our wastewater treatment plant spend something along the lines of $4 to $6 million a year in precipitating out that phosphorus so that we're not discharging it to the Detroit River, and thus causing or helping enhancing the ability for algal blooms to exist. So there's a lot of stuff. So triple bottom line framework is excellent at identifying non-correlated criteria such as social, environmental, financial. I suggest there's a few other components there that aren't captured in social, environmental, and financial, but also risk. But that triple bottom line framework doesn't include an inherent decision-making process, and a decision-making process is necessary for determining trade-offs. Next slide, please. There's 3 methods that are used there. Oh, Jamie's saying I need to be quiet here quick. Analytical hierarchical process by Sadie, which is a method of comparing different choices. Data development analysis is a free weight method of analyzing the efficiency frontier. And there's a few others, and there's also some details about each one. And then let's go to the final slide. I think this is my final slide. Limitations. Actually, let's go to the next slide, the very last slide. Is that the recommendations. What I suggest when you guys are looking at an environmental problem, you're using a decision-making trade-off mechanism to help bring all the stakeholders to the page. It is critical that you're hearing from everybody and actually analyzing everything when you're moving forward with an actual process. And there we go. So thank you very much.
Thank you, Dr. Norton. We will hear next from Dr. Victor Carmona on fecal indicator bacteria responses to invasive plant species. Thank you, Victor.
Thank you. The field data in this study stems from a course-based undergraduate research module on integrative biology that's based at the University of Detroit Mercy, but was made possible with support from Healthy Urban Waters, Dr. Vasquez, and Dr. Miller. Next slide, please. Our study site was Lake St. Clair Metro Park, which in 2013 had its massive 17-hectare parking lot transformed with a $3.3 million GSI project. On the left-hand side, you can see that the project reversed the historical drainage of the parking lot, redirecting it through a network of rain gardens. We knew from the primary literature that the invasive plant species can alter bacterial communities. Our focus was on fecal indicator bacteria, FIBs, which serve as a proxy for detecting the presence of pathogenic bacteria in the environment. Our objective was to study FIB changes throughout the rain garden network, but also with respect to the 2 invasive species that are common in the rain gardens, and you can see those on the right side of the slide. Our sampling took place the first week of November, so this marks the FIB dynamics at the end of the growing season. Next slide, please. The graphs on the right show FIB changes with respect to the rain garden sectors, which are identified on the figure left. As you can see, S1 and S8, which are the principal rain gardens draining the parking lot and golf area, had the greatest levels of E. coli. However, all sites scored statistically at or above EPA limits. This suggests that during the late fall, the rain garden network cannot sustain FIB levels below EPA limits for recreational water. Next slide. These graphs show the FIB changes with respect to the presence, absence of Phragmites, one of the invasive species. While mean levels of E. coli and coliforms didn't differ with respect to Phragmites, on the top left, you'll see that the variability of E. coli was significantly more consistent where Phragmites was present. At these locations, FIB scored statistically at or above EPA limits. This suggests that invasive Phragmites plants may actually be playing a role in reducing the variability of E. coli populations. Next slide. These graphs show FIB changes with respect to the presence of frogbit. While mean levels of E. coli and coliforms didn't differ with respect to frogbit, again, if you look at the top left, you'll see that the variability of E. coli was significantly more consistent at sites where frogbit was present. At these locations, E. coli scored statistically below EPA limits. However, coliforms scored statistically at or above. These findings suggest that invasive frogbit plants may also play a role in reducing variability of E. coli populations. It's important to note that GSIs are not natural systems, they're anthropogenic. These invasive plants may be recovering an ecological process that has not yet been identified. And because E. coli are harmful to humans, the definition, or by definition, the recovery of ecological processes by these two invasives represents an ecosystem service that merits further study as well as identification. Thank you.
Thank you, Dr. Kimona. Next, we'll hear from Jamie Stees-Thorsby on understanding flooding in Detroit.
Thank you so much, Barb. I am a program coordinator with Healthy Urban Waters, and I'm going to be talking about two projects, two pilot projects that we've been working on in the last couple of years related to flooding in Detroit. Before I get started into talking about the projects, I just want to really briefly talk about green stormwater infrastructure in case you're not familiar with it. It's basically put in to capture stormwater runoff or manage stormwater runoff. It replicates a natural system to collect stormwater, filter pollutants, and slow the movement of water into the stormwater system. And you can see some examples here. So the ways that is applicable in this project is basically what we did. We did this pilot study in a Northwest neighborhood in Detroit, and we basically created this model. Sorry about that. We created this model to analyze the before and after of how green stormwater infrastructure, if it's put in theoretically, how it could potentially reduce flooding in that area. I don't want to go into too much detail, but some of the results found that we actually, if we placed it in specific locations, there was some situations where it could actually mitigate flooding, especially if it was on a larger scale, such as putting in bioretention basins or putting in rain gardens across the neighborhood. The next project I'm going to talk about, that was actually inspired by the previous project that we worked on. The work on this study actually started because there wasn't a ton of information out there. We wanted to know about flooding within the city, and there was a lot of sources that had a lot of information, but it wasn't really thoroughly analyzed. So we began a pilot project to better understand within the city how flooding is affecting neighborhoods. We started out with a survey in Jefferson Chalmers neighborhood, which has within the last couple of years had a significant amount of flooding. We've compiled and analyzed household flooding data from multiple sources, including the survey data that we took, as well as survey data from University of Michigan, the City of Detroit, and the Wayne State University Center for Urban Studies. And then we came up with results and held a stakeholder meeting with community members, organizations, government groups, to discuss the results of these studies and to discuss how best to go forward. Just a really quick overview, some of the results indicated that renters and then specific locations had much higher odds of having flooded homes. So you can see on the map on the left here, the red areas show areas that houses were much likely to be flooded. Homes were much likely to be closer to other flooded homes. That's in the red. And the opposite is with the blue, shown in the blue. These areas, there were houses that didn't have flooding were much more likely to be near houses that also didn't have flooding. But if you want more information about this, feel free to reach out to me and I can provide you with some journal articles that have been published or will be published in the future. Thank you.
Thank you, Jamie. Our next speaker will be Alex Hamba, environmentally relevant PFAS exposures.
Okay, thank you for having me. My name's Alex. I'm a PhD candidate in Dr. Tracy Baker's lab. You may notice our water logo. This stands for Warrior Aquatic and Translational Environmental Research. Today I'm going to talk about why and how we use zebrafish to study the pathology of low-level PFAS exposure during development. Next slide, please. Since you're all at a symposium on water, many of you may already be aware of what PFAS are and our general risk of exposure. For the uninitiated, PFAS, or per and fluoroalkyl substances, are a class of highly useful chemicals. They're found in everyday products and emergency materials. They repel water, quell fires, and keep our eggs from sticking to the pan. They can do this because of their unique surfactant properties combined with their long chains of extremely strong carbon-fluorine bonds. These bonds, being resistant to degradation by heat, water, or virtually anything else, make them a very valuable tool. But at the same time, this means they stick around in media we don't want them to, such as drinking water or our bodies. We're mainly exposed via consumer products, contaminated food chain, or sometimes drinking water. Every single person in the industrialized world has a bit of PFAS in their system, and almost every tested water supply also reveals some PFAS. High doses of PFAS have a link to cancer, high cholesterol, thyroid issues, and pregnancy-induced hypertension. What does a low-level exposure, the kind most of us have, mean for our health? Next slide. First, we wanted to define the levels we may be exposed to in Detroit and the surrounding counties that receive their water from Detroit. You heard a little bit about this project on the slides from Dr. Baker. We chose sites along the Huron to Erie corridor relevant for public health and recreation. We found very low levels of the two most common PFAS, PFOA and PFOS, in the water at a metro park and downriver of the wastewater treatment plant, in a water body which goes on to be another state's drinking water downstream. Higher levels are found in the sediment, which small organisms can feed on and we're potentially exposed by eating the fish that eat these creatures. We also found higher levels of other types of PFAS, but chose to focus on the two most common. Next slide. Once we understood the environmentally relevant levels we're dealing with, I exposed embryonic zebrafish to low levels of PFOA, PFOS, and a 50-50 mixture of the two chemicals for the first 5 days of life. You've already heard from others in the Baker lab about the usefulness of zebrafish as a toxicology model. Since PFAS can cross the placental barrier, it's early life exposure. During this time in development, all major organs materialize. We then screen the larvae for physical abnormalities such as edema and lack of swim bladder, behavioral deficits, and aberrant gene expression. Next slide. What we found is low-level PFAS exposure increased physical abnormalities, changed their behavioral response to light, which can be an indicator of neurological health, and increased expression of genes involved in pathways of response to toxic chemicals, injury, and cancer. Clearly, PFAS is not inert in the body. Our next steps are studying any effects on reproductive capacity as these fish reach spawning age and their tissue-specific gene expression landscape in order to understand the mechanism PFAS works through. This way, we're closer to knowing the risk for aquatic life in humans from the ubiquitous PFAS chemicals. Thank you.
Thank you, Alex. Kate Ecuador is going to talk about tree canopy and water quality improvements in urban areas.
Thank you. My name is Kate, and my research is focused on tree canopy cover and water quality improvements in urban areas. Next slide, please. So I've summarized my objectives of this research as follows. How has tree canopy cover impacted stormwater management? How does the introduction of trees into stormwater control measures increase their retention capacity? And how do we increase tree canopy cover in areas lacking such coverage? Next slide, please. An article published by Climate Central Organization in 2020 tells us that 400 billion gallons of stormwater are managed annually in the United States. And trees do this by using their leaf system to hold on to a percentage of the precipitation that would normally fall on the ground. Trees are known to have extensive root structures that also enables them to hold on to another percentage of precipitation that would normally fall on the ground and generate some water runoff. And in addition to these, trees' root system creates preferential pathways that also increases the infiltration capacities of soils. Next slide, please. It is also important to note that if we're referring to trees availability in the city of Detroit, American Forest Organization reports that the tree canopy in Detroit is between 18 to 22% compared to the tree canopy of the United States as at 2004. But one thing we should know is that the maps you see on your right-hand side are maps that I got from the Greenway of Detroit. They were created, the very first map was created as a result of an inventory that was tree planting that was done in 2016, while the bottom map is a map that I got from the American Forest Organization. What is obvious from both maps is that even though it's stated that canopy cover is between 18 to 22%, this is not evenly distributed. You will notice that some districts seem to lack tree canopy cover. Next slide. Thank you. In summary, it is important to know that a city's tree canopy cover should be determined by its goal, its target goal. This goal explains what the city plans to achieve. For example, if a city like Detroit wants to manage more stormwater runoff, then they have to design plan their tree canopy cover to meet that target goal. And also, it's also important to know that for city managers, there is a need to collaborate with the residents to achieve whatever goals they plan to achieve. Thank you.
Thank you very much, Kate. We'll hear next from Amir Kamju on groundwater intrusion to the regional wastewater collection system.
Hi, everyone. My name is Amir, and I'm going to talk about the intrusion of the groundwater into the sewer system in the Detroit metro area. Next slide, please. So the sewer system lines are affected by many factors leading to defects by the time. We have exfiltration when the sewer leaks from the lines to the ground, and we have infiltration when we have higher groundwater elevation. So the groundwater is going to be intruded to the sewer system. Infiltration has many economical and environmental drawbacks, including the excess pumping, treatment, and operation costs, soil wash-off, ground surface damages and sinkholes, sewer system deteriorations, and also sewer backups, combined sewer overflows, and groundwater contamination transport by the pipes. So as we're talking about One Health, let's go to the next slide and see how contaminated this is land. In these maps that are provided by Eagle, you can see the landfills, the contaminated sites, and the leaking underground storage tanks. To focus on one of those contaminated sites, which I hope we remember that, let's talk about the green ooze coming out from 696 in Madison Heights, which was containing the chromium-6 and PFAS contamination, which are both toxic and causing cancer. So if you look at the schematics, the contaminated groundwater was placed 4 feet below the ground, where we had storm sewer submerged, and also the groundwater contaminated groundwater found its way to the storm sewer in the highway. As one of the first things that they did in this $3 million project of remediation, they lined the sewers to make sure there's no infiltration to the sewer lines. And, you know, we have many sites like this, which are known, and many which are potentially unknown. So this emphasizes on the importance of the study of the infiltration to the sewer lines in Detroit City. Thanks.
Thank you, Amir. We'll hear next from Dr. Chai-Chun Wu, point of use carbon block drinking water filters.
Thank you for having me. I'm a postdoc fellow working with Dr. Tracy Baker. Next slide, please. Today, I'm talking about commercial drinking water filters that are used to remove disinfectant residual to get better taste and odor, or remove metals, industrial pollutants from contaminated tap water. So these filters have been deployed in places such as Flint, where there were chemical water quality concerns in the tap water. However, these filters actually can produce higher bacterial concentration and very different bacterial community in the filtered effluent compared to the tap. Next slide. So we want to better understand the impact of the filtered drinking water on human health. So we use a Zebrafish model to evaluate the changes in gut microbiome development and behavior after the zebrafish were exposed to water. Those were collected at beginning or the end of the filter age. And adult and larval zebrafish were exposed for 14 and 5 days respectively. Next slide. So this slide shows the bacterial composition in infant, effluent, and the gut microbiome of adult and larval fish over filter age. So you can see each color in this bar plots indicate the relative abundance of bacterial family in the sample. Overall, we found that water and gut microbiome has very distinct bacterial composition with each other. At the left-hand side of the slide, you can see that the effluent water microbiome changed more significantly, more apparently over time than the infant. And correspondingly, if you see the up right corner, the gut microbiome of larval fish exposed to the effluent also had more apparent changes between the two filter ages than those exposed to the infant. So at the down right corner, you can see the adult microbiome exposed from infant or effluent were actually consistent over time. So next slide. So following the larval fish exposure test, we screened for abnormalities during larval development and assessed their locomotion behavior, responded to dark and light stimulus. At the left-hand side, larval exposed to effluent from two filter ages had similar abnormality rates in various phenotype endpoints. But at the end of the filter age, larval exposed to tap water had more abnormalities than the control and filtered effluent. So for the behavior response, we show at the right side of this slide that larval exposed to effluent at both filter ages had higher locomotor activity compared to the control. So this indicates a neural behavior changes were induced by exposure to filtered effluent. And there are more and more studies discover gut microbiome can affect neural health in humans. So we are working on understanding how bacteria in gut alter zebrafish neural behavior and how filter affects larval gut microbiome. And hopefully this results can be translated from zebrafish to human health. And we'll have better understanding of the health consequences of using these drinking water filters. Thank you.
Thank you, Dr. Wu. We will hear next from Brendan O'Leary on the Detroit Subsurface Characterization Project.
Thank you. I'm a PhD candidate working under Dr. Miller and with the Healthy Urban Waters program here at Wayne State University. Let's read my slide here. So my talk today is going to be talking about two field sites that I'm currently developing to characterize the subsurface and evaluate VOC movement in urban landscapes. And this is a partnership that I have with EGLE, the Environment, Great Lakes, and Energy Department here in Michigan. And specifically, I'm working with Beth Benz, who's been a great project manager and collaborator on this project. Our two field sites is one is a leaking underground storage tank on the east side of Detroit, and that's pictured here in the center left of the slide. And on the right side, there's an old dry cleaning facility that has a significant amount of TCE located underneath the building. You can go to the next slide, please. So VOCs are a group of aromatic or chlorinated organic compounds that have a high vapor pressure and low water solubility. And they're a common pollutant in the environment, especially in the subsurface. The map in the top center here is all the leaking underground storage tanks located throughout the state of Michigan. And you can see these are fairly concentrated in urban centers. The map in the bottom right is a map of all the leaking underground storage tanks in Wayne County. And again, these tend to be concentrated along major highways or population corridors. And VOCs pose health threats, both short-term and long-term. And one of the exposure pathways that I'm evaluating is vapor intrusion into houses. You can go to the next slide. So these again are my two sites at Conant. This is the dry cleaning facility located just north of Hamtramck, and the Strong site, which is my leaking underground storage tank, which is an old gas station. This is just east of the old Hamtramck assembly plant. And to evaluate VOCs in the environment, I'm using a number of different approaches that include soil sampling, groundwater sampling, soil vapor sampling, and kind of a new emerging approach of using vegetative sampling to evaluate the bounds of the underground contaminant plume and understand the concentration gradients throughout the site. Go to the next slide, please. And why this is important, I mentioned vapor intrusion earlier. The model here shown on the right is an example of how groundwater will transport the underground VOC plume, and then over time it will volatilize and move through the unsaturated zone and ultimately into human living space of a basement, or if the house has a sub-slab, it will move directly into the first level of a house. And urban changes can impact this movement, which include sewer systems, as shown on the example on the left side here by the green lines, or potential drainage pipes or other types of preferential flow pathway that can alter the VOC movement on the subsurface. Next slide. So right now we're working in the field to understand the subsurface and take some samples. Here's just some example pictures of the WSU geophysics class working out in the field on the top left. Dr. Glenn Hood and my lab assistant, Kelvin Saligan, working in the top right. We recently had a geoprobe out there taking continuous soil boring samples earlier this month, and the Conant site is shown on the bottom right, so thank you.
Thank you, Brendan. Our next lightning talk comes from Dr. Jeff Ram, talking about bacterial diversity and potential sources of contamination at metro parks, or the beaches, excuse me.
Next slide, please. The idea of One Health is that everything we do and the environment around us can contribute to the overall standard of our health. We want recreation because the outdoors feels good and the exercise is good for us. And that includes swimming at the beach. But will it be safe? Each summer, our local health departments measure E. coli in beach water to determine whether it's safe to go in. Some of our local beaches sometimes sound like petri dishes. High E. coli levels today, stay away. But really, in the Detroit River and Lake St. Clair, how much bacteria is there and what kind is it? Where does it come from? And most importantly, is it dangerous? We collaborated with the University of Windsor to measure bacteria at two local beaches, Belle Isle Beach and Petri Dish Point, Sandpoint Beach in Canada, Windsor. Next slide, please. Measuring E. coli almost daily, we found that on most days, the E. coli levels were at safe levels, below 130 CFU per 100 ml on a 30-day moving average, except if we collected water close to shore at Belle Isle Beach, where we saw a rising trend from July through September. When we used high throughput sequencing to measure not just E. coli, but the diversity of all bacteria at the beaches, we found some of what we expected. First, that the mix of bacteria changed across time. Over on the right, here at Belle Isle, the mix of bacteria in June was much different from the mix in August. You can see separate clusters there. Indeed, hardly any overlap in a variety of diversity measures. At Sandpoint, it was similar. June was much different from August. July was in between. What wasn't expected, though, is that the diversity at Sandpoint had almost no overlap with the diversity at Belle Isle. The clusters of sequences were completely separate from the clusters at Belle Isle. While many genera are the same, the proportions differ greatly between the two beaches, regardless of the month. Next slide, please. Where do all these bacteria come from? The high throughput data had enough information to compare to fecal bacteria from various source animals. A previous study that we did at Lake St. Clair Metro Beach with source indicator sequences in E. coli had concluded that birds accounted for about 50% of the E. coli. An analysis by the high throughput methods in the recent project reached virtually the same conclusion, about half from birds, only about 10% from humans, i.e. sewage, at both Belle Isle and Sandpoint. Next slide, please. Is it dangerous? You can't read the names of the bacterial genera on this list. It's too small to read. But this is a list of the genera that were most highly correlated with high E. coli levels at the beaches on those days where it was high. The boxes, since you can't read it, I've boxed in the genera that are known to include pathogenic strains. At Sandpoint, at the top, these include Pseudomonas and Legionella, whereas at Belle Isle, in the deeper water, the top 5 correlates include only Pseudanocardia. Whether these are the toxic strains of these genera are unknown, but this is the direction of research for finding out how we can maintain One Health. And finally, I'd like to give thanks to Healthy Urban Waters for support of this project.
Thank you, Dr. Ram. Next up is myself. I'm going to discuss the development of the Fatberg game. And I'm a project coordinator under Dr. Miller with Healthy Urban Waters. First slide, please. In the summer of 2019, Lawrence Tech Game Arts students were looking for a summer project. And through Healthy Urban Waters, brainstormed with Wayne State research students at the Water Lab to bring visualization to environmental issues. At this time, the researchers were investigating the fatberg that Macomb County was experiencing in the sewer systems. If you don't know, it's this humongous blockage in the sewer systems due to solidified oils and waste. Most of the Lawrence Tech students had not heard about this at all, but they pounced on the idea of creating a game that would educate about the fatberg and what we need to do about it. Thus, Don't Feed the Fatberg game came into being. Next slide, please. The approach was the combined LTU and Wayne State teams set out to create a serious game that's based on education, not on entertainment. This type of game moves the player from an engagement to acquiring knowledge, to taking action, and actually changing behavior. Next slide, please. So in order to create the game, the students used an agile execution strategy, which is a software development strategy that engages the researchers all along the process of creation of the game. They're involved in testing and validation of many of the steps. And it's a methodology that's been proven to increase accuracy and quality of software development due to the involvement of these stakeholders. And on the right-hand side, you can look at the large team, the work breakdown structure that's involved in actually putting a software game together. Most importantly, you need artist skills, game design skills, software development skills. The artists actually create the characters, the trees, the flowers, the houses, the environments. The game designers actually put together how the game flows, its levels, and how the player progresses through the game. And then the software developers have helped to code these actions and put it into production. Next slide. So this is the results of what was developed by the Lawrence Tech students in cooperation with the researchers at Wayne State. The game starts out in your apartment or house, and it's a mess, so it needs to be cleaned up. There's lots of stuff on the floor that should be either thrown away or thrown into the toilet. You have a choice of throwing it there, of throwing it down the sink, or into the waste base receptacle. And to note, all of the objects on the floor were actual things that have been found with the fatberg that the researchers uncovered. At some point, the fatberg gets too big, so you have to go down into the sewer system to figure out what's been captured by that fatberg and eliminate it. So at this point, you are searching for what is has been captured by the fatberg since you put it down the toilet or into the sink, and you pull it out and then figure out what the issue is around why you should or should not put it into the sewer system. And so you decode at a second point of the game and figure out what the issues were around the way to dispose of it. So very, very simple, but that's what they came up with. So thank you very much, Jamie. Oh, I think it's me. Up next, we will have Theo Fowen talking about ecological rehabilitation at Eliza Howell Park.
Thank you, Barb. And I'm very impressed by the fatberg game. First slide, please. You can go to the next one. So, hi everyone, my name is Faye Fowen. I'm the Director of Green Infrastructure at the Greening of Detroit. Many of folks are familiar with our community tree planting program. We also install green infrastructure. So in this project, what did we do? We installed a shallow green GSI practice using native plants to manage 112,000 gallons of stormwater runoff per rain event from a major highway, I-96, actually the service drive, as well as the surrounding park area. The project, if you can click next. Oh, okay, we can keep it there. The project is located in Eliza Howell Park, which is the city's fourth largest park at 250 acres in Northwest Detroit. It captures, like I said, surface flow and stormwater runoff. These photos show here the installation of that project. In that previous image, there was using heavy equipment excavating the site. Next image, seeding and seeding the site with native upland and wetland species. And then this image right here shows us doing the erosion control blanket afterwards. It's in the wrong order, sorry. But with all of our work, community engagement is a big part of the work. And so whenever we're doing GSI or tree plantings, we make sure to include members of the community and external volunteers to show this work being created. Next. Yep, and that's where the park is located. Our partners here were the City of Detroit, OHM Engineering, as well as our community partner, Sidewalk Detroit, who programs the space, but also was a lead on helping create the master plan a couple of years ago. Next slide. So planting design for stormwater management was a very big feature for this project. We work closely with the engineer and the contractor. Next slide. Click. And one more. So as a result of this work, we removed 13,000 square feet of old asphalt parking lot. reclaimed about tons of limestone aggregate to create a pathway within the park, installed 50 native trees, 144 shrubs, and native seed mix with volunteers. Next slide. And that is one of the trees that was planted, witch hazel, last fall. Next slide. Ecological rehabilitation was a big part of this project, and so that allowed us to identify species that could do well in this ecosystem. Next slide. All right, so basically we did a lot of research. We compared resources from Michigan Natural Features Inventory, which you should check out if you haven't before, to understand what was existing in the landscape before settlers came, as well as the 2004 Flora Report of Rouge Park. So with that, we were able to choose different tree species that should manage well in this space. Next slide. And this is my last one. So basically we've installed the site, and now we are looking forward to establishing the vegetation here. It's very important we do green infrastructure that looks good at the end of the day. Thank you.
Thank you, Feo. Now we turn it over to Jamie for questions and answers.
Yeah, so if anyone has any questions for our lightning talk speakers, please send them to me right now in the chat, and I can read them off, and then we can highlight whoever, or spotlight whoever, whoever the question is directed towards. And then Bridget will also be helping me with this.
I think, can you hear me?
Yes.
I think Paul Kilgore has had to leave already due to another commitment. So while we're waiting for some questions, we
actually just got one.
Oh, great. Okay,
question.
Go ahead.
So this is for Faye. What was the name of the app that you discussed about native species? I'm assuming that's the app that's about native species, like originally in Michigan.
Sure, I can send you the link, Jamie, but it's the Michigan Natural Features Inventory, and it is managed, it's by the state of Michigan. Really great resource. They have they have maps of every single area within the state of Michigan, and I've been able to interact with their staff for questions on how to plant specifically around the hydrology within this ecosystem.
Great, very cool. Yeah, if you could send that link, I would love that, and then I can post that for anyone who's interested. Okay, sorry, Bridget, I interrupted you when I got a question in.
That's okay. So I have a question for Danielle Meyer. She presented about the nanoplastics and zebrafish. So I don't know, can we spotlight her?
Sorry, could you repeat that?
Yeah, so based on your findings of dysregulated nervous system development and energy metabolism in the zebrafish model, what are the potential implications for wild fish developing in plastic contaminated environments?
Sure. So since we're seeing effects on the neuromotor systems and energy metabolism, we're thinking that can affect the overall activity level. So that will affect their ability to find food and also evade predation in the environment. And also we're thinking that we could see this later on in life affecting the reproductive capacity, and then maybe just generally activity and behavior levels. So there's a lot of different directions we could look into for this.
Yeah, thank you. That's really interesting.
We got one.
Oh yeah, go ahead.
Okay. One question for Jeff Ram. How did the sampling techniques vary between Canada and the US sides? Is there any difference between them, or any different methods that are used?
We used identical procedures. The University of Windsor did the quack basically, and we basically said we're going to sample at about calf deep and waist deep, so that's the near shore, and deeper water sampling. And we did quantity measures for E. coli. For the high throughput sequencing, we captured bacteria on filters, kept those in the freezer, and then sent them to the laboratory at the University of Windsor, where they did ion torrent next generation sequencing to describe the multiple types of bacteria that were included. The paper's in preparation, has been for a while, but I think within a week or two, it's going to be finally submitted. Yeah, that's the answer to that question. We used the exact same techniques, and the high throughput sequencing was all done at the University of Windsor.
Okay, great. We have another question. This one is for people just in general who are doing research with COVID. So is there any contribution from animals infected with COVID to wastewater? Could that could potentially be due to combined sewer overflows? And then what animals catch COVID and is it prevalent? So that would be to Dr. Schuster or to, I'm not sure if Irene is still on, but
we run the laboratory in Dr. Schuster's project. We do the measurements.
Oh yeah, so Jeff, you could also answer this too.
Yeah, I mean, Jeff and I can tag team on this, but briefly, a lot of at least the prevailing research results indicate that the SARS-CoV-2 viral material in the wastewater media is deactivated, that it's not particularly infectious. Jeff, would you?
Well, I just add, unless people are dumping their the dog feces that they pick up when they're going around walking their dog down the toilet instead of putting it in the trash, it's not going to appear in the wastewater stream.
Unless there's a combined overflow.
Combined sewer system or storm sewers?
That could certainly happen. That's one of the reasons why, well, Bill could talk to this about where we're collecting the water for the dorm project. I don't think combined sewer affects that, Bill.
Well, that's, as Irene pointed out, when we have both an opportunity and a set of constraints when we look at the wastewater collection and conveyance system as a place to find the SARS-CoV-2 viral material, which again is largely inactivated in the digestive and excretory process. So the dilution and the actual, what are the sources that are feeding into a particular sewer shed? And, if we had time to go back to the results that I presented, we have to be pretty specific about tracing, understanding which pipes lead where or collect from which dorms, for example, in our case, and take a look at the hydraulics and the hydrology and how that affects what we might be seeing and making sure that they correlate. So it's a bit long-winded, or I'm not sure if that gets to your point, but that would be my response. But further, this is part of the excitement of wastewater-based epidemiology, and we're pretty good at these things, and there's good modeling resources. It's oftentimes that the master plans for the buildings that we work in and the populate our built infrastructure, they're not those, the as-built can vary quite a bit from the what the designers thought would be built. So
I'll just add one thing. The dorm project, those samples were being collected from the drainage of the dormitories probably before any combined sewage entered. However,
right.
We were sampling also some sewer sheds that included the hospitals. And when we had snowy weather where you might expect salt to be on the roads, we saw an increase in the conductivity of the samples we collected, suggesting that there is combined sewage input to those samples. And so it would be possible if people haven't picked up, let's say, dog feces, that would be a small part of the signal. But primarily, when you're getting sewage at the treatment plant, you're getting, I would say 99.9% human fecal matter and very little from pets.
I guess I would be happy to speak a little to the question of animals affected by COVID, just briefly. There's several species known to be able to be infected by COVID. I think right now one of the biggest concerns in general are mink, just because it seems like they are getting infected with COVID and a new viral strain is being generated within mink populations. In general, in terms of our domestic animal species and even zoo animal species, we've seen some tigers test positive at various zoos in the US, for example. Those incidents are fairly isolated and rare still at this point. So yeah, I agree that in terms of wastewater signatures, it's probably minimal, if that.
We have a few more questions related to COVID, the COVID testing projects. So for Dr. Schuster specifically, was your group able to sample near the Anthony Wayne Drive Apartments, which is being used as the quarantine facility? And if so, what were the results?
Yes, and at this time we only have 2 event dates. You have to understand that in this pilot project, we ran up with the RAM lab in the School of Medicine. We basically stood this up in about a month, and that's integrating new technologies like the ddPCR instrument. And so we only have 2 event dates analyzed this time. But by the end of probably around March 1st, March 15th, we'll have a complete time series. And so what we'll be able to do, we'll have a complete time series that is for each of the sampled drainages or sewer sheds. We had 2 in the greater downtown area, and then I think we had 5 dorms, various points in their drainage system. So what's exciting is that we're going to have a very nice time series here, and then a very precise census for quarantine and isolated individuals. And again, this is anonymous because we're sampling population level SARS-CoV-2 signal in these drainages. And I hope to get with Irene about fitting our data to her exposure risk model for SARS-CoV-2. I think that'd be very productive. But again, by using time series analysis, we can start to unpack, the strength of different factors, be they environmental, like water quality parameters, or actual numbers of individuals in isolation or quarantine. It's a pretty neat study. Hope to keep going with this in a post-vaccination epidemiological environment.
Yeah, absolutely. And then we have one more question about the COVID project. So after you saw preceding spike of COVID in wastewater, can you reveal that what are the measures the university or other agencies are doing to deal with the potential outbreak?
So for better or for worse, this is a retrospective study, and we are working closely with the Public Health Restart Committee here at Wayne State that involves everybody, housing, facilities, the police, our combined talents as a faculty. And so what has happened here is one thing that I think we have confirmed is that the isolation and quarantine policy has worked very well. We have not had any sort of large outbreak here at Wayne State for some time. We are dealing with, a large, not as large as it could be, but about half the population that we would expect in ordinary times in our dorms. And so through a lot of, it's taken a lot of hard work from especially our housing and facilities folks to really get a handle on this and be very clear in their enunciation and implementation, isolation and quarantine.
Well, okay, well, we're gonna wrap up the question and answer at that point. Let's go on to, first we want to thank all of our speakers at this event, and we really appreciate everyone's research and the ability that they had to present their research. It's really cool to see all of these projects, especially I know most of you and I work with most of you, but I don't know a lot about some of the projects that you presented. So it's really cool to see it, even for myself, see what everyone's working on. The next thing I want to mention too is some of our upcoming events. We have a number of water at Waynes this semester that I haven't sent out the marketing information on because I'm still finalizing everything, but we have one coming up on February 11th and then one in March and two in April. And a few of them are related to COVID. So definitely the hot topic here. Yeah, so I highly recommend, I'll be sending out more information about that in the next week or two. But I highly recommend if you're able to attend those, I would definitely recommend doing so. I also want to say too, this is being recorded and we are planning to post this on our website. So if you want to refer back to something or watch this again, feel free, or pass it on to someone else, feel free to do that. We would love to get this information out to as many people as possible. So now I'm going to actually pass it off to Dr. Carol Miller and Dr. Runji Morris to talk about the One Health Initiative kickoff.
Thanks very much, Jamie and Barb, for sort of keeping us going on this symposium and keeping everything on time. It went very well. It was exciting to hear about everybody's research. Just like you said, Jamie, some of it I knew about and some of it I wasn't very familiar with what people were doing. And that's one of the collateral benefits of this, is that hopefully it initiates even more collaboration here on our own campus, as well as with some of the other universities that we've heard from today. So for those of you that are interested in learning more about the Integrative One Health initiative that's being kicked off here at Wayne State University, we welcome you to stay for this session. I think it's especially applicable to Wayne State University faculty and student researchers. But we're, again, just in the collaborative nature of things, we're looking for community partners and other research partners that are interested in these topics also. So everyone's welcome to stay for this session. It's relatively short, but just to give you a little bit of an idea of this initiative that's being kicked off. Before I get any further, I wanted to introduce my trusted colleague and friend, Dr. Melissa Runga Morris. I've had the pleasure of working with Melissa now for probably more than 6 years. And I think that my introduction to One Health might have been through the collaboration that I started with Melissa 6, 7, 8 years ago, because before that, I was just a regular old engineer. And once I started working with Melissa and getting much more involved in the human health, medical side of things, it really opened up a lot of insights for me. So I would like to welcome Melissa, and I also had asked her to perhaps say a word or two about One Health and this new initiative, perhaps a little bit about the vision associated with this.
Thanks, Carol. I'm happy to talk today, and I think you're too modest and really got the thing the other way around, because it was from you I learned about the potential for One Health and how engineers in these times, the engineers aren't just putting up pipes, but actually looking toward maximizing human health protections in our urban environment so that we can be safe in the built environment long term. I'm a physician scientist. My name is Melissa Runge Morris, and I'm the director of the Institute of Environmental Health Sciences at Wayne State. And I also serve as the director of the Center for Urban Responses to Environmental Stressors. That's an environmental health sciences P30 core center funded by NIEHS. It's my privilege to be working in co-leadership with Carol Miller to launch the One Health initiative at Wayne State with very good and generous support from our Vice President for Research Office. Carol is a great collaborator, and I also want to recognize our inspiration in working with Dr. Bridget Baker, who's a very, very talented veterinary scientist and is equally passionate about One Health and the need for One Health in our Wayne State University community. In the Cures Center, what is it? It's a center that's supposed to catalyze research in environmental health, but how can it do that? How can it do that without looking through the lens of One Health? And we soon came to realize in Cures that you can't just consider environmental exposures and human health endpoints without really taking into account effects on animal health, plant health, ecological health, planetary health, when you look at the big picture and what our environment means to our future. So I just want to say thank you for this opportunity, and we are looking to do big things for the growth of this program and for the benefit of our urban community. Thanks, Carol.
Yeah, thanks so much, Melissa. I am going to kind of move through my slides now, and so I'm changing the view a little bit here, and I'm going to my own slide deck here. So I did want to say, and Melissa's already done that for me, but yes, the inaugural science coordinator for this One Health initiative is Dr. Bridget Baker, and you've all been introduced to her a little bit, but Bridget, you know, being the doctor of veterinary medicine among us, perhaps you'd say a few words about your perspectives of One Health before I take this any further.
Sure, thanks, Carol. Well, yes, as a veterinarian and former wildlife biologist, obviously animal health and welfare and wildlife population health and conservation are kind of my main interests, but in working with animals in various settings throughout the years, not only is it obvious that environmental health is essential for animal health and is also dependent on people, but I think more people, especially with the COVID-19 pandemic, are appreciating that our own health is also very much inextricably linked to wildlife health and ecological health. So I'm really excited about the One Health initiative moving forward. You know, Wayne State has major strengths in human medicine, biomedical research, engineering, and environmental science, and I'm really excited to bring kind of the veterinary medicine and animal health perspective to the initiative, and to hear more about what the initiative is hoping to achieve. So thanks.
Thanks very much, Bridget, and thanks for agreeing to take on this new opportunity. So the meaning behind One Health, I think, is likely familiar to everybody who's on this call, but just, you know, there are a lot of different ways of looking at One Health, and it seems like different organizations have different definitions or descriptions of One Health. So I thought I'd just put up here from the Centers for Disease Control, their sort of succinct description of One Health. And I think it's very similar to what Bridget just mentioned for us. But the idea here is that we want to recognize the health of humans being closely connected to the health of animals and our shared environment. So I guess I'm, I look at myself in terms of how I fit in this One Health picture. And, years ago I would have said, well, I only come into here in the environment because I'm an environmental engineer. So I know something about the environment and that's my segue in here. But the more that I've learned about it, I've learned that, yeah, I am an environmental engineer and I do bring that expertise, but gosh, I'm a person too. So I have a little bit of expertise about people. I'm a parent, so I've learned about health of my kids as they grow. So I have hopefully a good amount of expertise in that area. I'm an animal lover. I've had many animals in my life. So I guess, in addition to the concept being very holistic, I kind of look at it as we as individuals are also quite holistic, and we all bring a variety of insights and perspectives into this. And I think by opening our head up a little bit that way, I think that there are many more advances that can be made. And so that's just my own personal take on this. So in terms of more specifically One Health at Wayne State University, this is still sort of fluid. As Dr. Lanier mentioned, this was kicked off formally on January 1st. And so we're only about 20 days, 25 days into it here. And so we're putting structure behind this initiative as it is right now. But there were a few key pieces of understanding that went into the solidification of the agreement between Melissa, myself, and Dr. Lanier in the Office of the Vice President for Research regarding what the One Health Initiative is. So for those of you that are interested in getting involved in it, I think it's very useful for you to see what some of these ideas are that we actually put down on paper. So Melissa and I are going to be responsible for coalescing environmental health sciences research activities and enabling technologies across campus with strategic partners to target themes and thrusts, with the output being direct relevance to our community and also additional external funding. This shouldn't be something that forever is always funded by Wayne State University or through contributions from the Earth Family Foundation or others, but of course we want to go beyond that, and that's our plan. We also have an agreement with the Office of the Vice President for Research that we will be looking at the involvement of the One Health theme in future faculty recruitment opportunities. And I think that's very important because that hasn't really been a specific theme of faculty recruitment. And this can impact colleges and departments across the university because it is not simply a civil engineering or med school or pharmacology or law. It's, all of those play a role in One Health research and also the academic side of it. We do have an agreement with the university to come back and look at this One Health initiative in terms of, does it continue as an initiative or is it mature enough in a few years to evolve into a center? So I think that for those of you out there that are really interested in One Health, I think that should give you some something to smile about. It may be something that is actually going to be available as a full-blown center. I think it's also important to see this last point here, that we will be working with the Office of the Provost to develop training programs in this broad area of One Health with environmental health sciences. So that was kind of the beginning as we're starting to put structure into this. We will be developing an internal steering committee and an external advisory board that has not been completed yet. Hopefully some of you who may be listening to this will become part of one of those. One of the things that the One Health initiative will enable, now that it is kicked off, there is some money associated with it. So one of the things we will be doing with a portion of those funds is to support pilot projects that are specifically, that have a specific interaction with this One Health theme. The kickoff of that is today. We really hadn't shared this idea with anybody until now in terms of this funding opportunity being available. And I can see that I'm coming in and out of my green screen here. But this funding opportunity is coming available today. So there is some written material or some actual material that will be sent to all Wayne State University faculty and students regarding this program. Some details are available, letters of intent. After we provide some information about this pilot project program today, we will be soliciting as part of that letters of intent. And we give information in some handout material we've developed that we will send to you via email. We give you information about how to submit letters of intent. They will be due in about 1 month from now. And these are relatively short, several pages. Those of you that have been involved with the CURES program that Dr. Runga Morris is the director of are familiar with these types of letters of intent. We will review those along with our steering boards, and we will notify successful applicants from the LOIs that they are given the green light to prepare a full proposal. Those full proposals will be due by April 9th, announcements of awards by April 30th. These dates may change slightly, but this is our schedule at this particular point. The amount of money that we're setting aside for pilot projects will be not to exceed $50,000, and those will all be $50,000 towards direct costs. Okay, and I think Melissa, I'm gonna, I'm getting a little bit dry-mouthed here. I think I've been talking way too much. Do you want to say a little bit else regarding the types of submissions we would be looking for?
Yes, I do want to also emphasize the fact that letters of intent and pilot project applications are reviewed by an external and internal review team. And please bear in mind that these opportunities are there so that we can grow the extramural funding base in this initiative for Wayne State University. So we're looking for projects that have high potential. In my case, it might be for NIEHS funding potential, but also relevant to One Health would be NSF funding related to the One Health initiative and other pertinent institutes. You heard today from the program, the range of the topics in this program indicate to you what One Health's all about. It runs the gamut from engineering concerns in the built environment in our urban city, but we're also looking at threats from infectious disease elements through the watershed. We're looking at just that intersection between environmental exposure and what informs our ecological health in our urban domain. So that it sounds quite complex. It also sounds quite broad, but anyone who listened today to this program gets it. I mean, yes, it's necessary to have a transdisciplinary team of scientists like yourselves involved in One Health concerns, because only that can give us the answer for better health in the future. So, so do bear in mind that this type of opportunity for seed money is not totally speculative. We're looking to grow projects that have a high potential for extramural funding in your institute.
Thanks, Melissa. So a couple additional items. The PI on the project proposal needs to be a Wayne State University faculty or student or staff. But certainly co-PIs, senior investigators, collaborators can be from other institutions, from watershed organizations, even consultants, if that's what makes a good team. But we do want the PI to be a Wayne State person. I think it's, we've just about used up all the time. It's 4 o'clock, but if there are a couple questions, I'd certainly be happy to try to answer them. Like I say, we're still in the formative phase here, but we wanted to take advantage of this venue where we had the symposium to also kick off the One Health program. And I guess if there's any questions, it would be fine if you wanted to just say them out loud or to follow up with questions to either Dr. Runga-Morris or myself. And you'll be hearing much more about this as we learn a little bit, as we develop the program a little bit more. It's been an exciting day, and it's great to have such a large number of really impactful projects, and most all of them dealing or focusing very much on the Detroit area. We know what our focus is here at Wayne State University, and I think that the range of presentations we saw were very much along those lines. If there are no questions, it's 4 o'clock, and that is perfect timing. I hope you all have a wonderful afternoon, and thank you all for taking the time to be here.
Thank you.
All right.